Okay, we are recording. So if anybody has questions, now is a good time to ask. I didn't want to take any questions while we weren't recording, just in case somebody missed it. I'll do some attendance. Let's see. Kip, David, Hodges, Sipple, Eric is online. Midkiff is online. Bryce is here in red. A little extra credit, plus you're here, so a little bit more extra credit. Cedric Richardson, physically here, a little bit of extra credit. Same with Massey, Newton, Burdette is online. Hudson, Felder, Tudor, Asbury, Bowers, Hemmings, Redmond, Reed, Kayla's here in red, a little extra credit on top of extra credit. Kenzie's here physically. And Van Cross. Did I miss anybody? Everybody's accounted for? All right, well, we have less than a minute. I'll go ahead and pull up this PowerPoint. All right, well, I'm going to wait till 8:01 to make sure everybody's here. So in the meantime, are there any questions? Is anybody going anywhere for spring break that's worth mentioning? No, me either. We have spring break on a different week than my kids have spring break. One announcement, I'll remind everybody again, read the announcements that I post. Just extra credit there. Some people just missing stuff. Like, how many of you knew the exam was on Wednesday? I recognize all you guys, so yes, it's all of you, and then some. Like, I keep repeating myself, and people keep messing up. Like, I didn't know the exam was on Wednesday. Or I didn't know we had to come in the lab. All these things, I keep repeating myself and sending you reminders. I'm just going to leave announcements. I'm here if you need help. Reminder, again, if you need a boost on a grade for a certain assignment, meet me online so we can talk about your assignment, talk about the questions you missed and why the right answers are the right answers and the wrong answers are the wrong answers, and you can learn from it and get a boost to your grade. I see Asbury is online. So yeah, you guys just pay more attention. I think a lot of it is because I don't know. I really don't know. Maybe because you're not forced to be here. So moving forward after spring break, I'm going to make it harder for people online. So I've tried you'll have to jump through more hoops if you come in, if you attend online. Because if you remember back when we first met on the first day of class, I said that the online option was there for, like, if you just weren't feeling good that day, you happened to slip in, right? So you had those options if there was something that came up in your life and you just couldn't come in. I didn't intend for it to be an everyday occurrence. And it's really showing with grades. So I just want to help you, especially before the drop date. Our affairs in order before then. So any questions before we jump into chapter 8, and then I can leave you for spring break. All right. Let's see. Let's do it here. So attendance. As usual, you have to send the words before 9 a.m. Anything past that is late. And if you're watching the video as opposed to the live stream, make sure you send it before the next time we meet for lecture, which in this case will be a week, so you'll be set. Anyway, here we go. Let's talk about meiosis, right? So Let me back up a little bit here, actually. Let's say the first word for attendance, just to start it off, will be reproduction. Reproduction will be the first word for attendance. And as usual, if you're in person, you can send me those words if you want for a little bit of even more extra credit. So yes, reproduction, sexual reproduction, as a matter of fact. Just to remind you, in the first bullet point, we just kind of talked about cellular reproduction in general, right? We said there was sexual reproduction versus asexual reproduction, and how asexual reproduction is performed or is accomplished by mitosis. Sexual reproduction is accomplished with meiosis, right? Then we actually talked about mitosis in the cell cycle. So now that we're done talking about mitosis, we're going to learn about meiosis. So the exam for this portion is going to be a lot of comparing and contrasting, right? So for mitosis, it's going to be a lot of what in this stage, this happens, and that stage, that happens, right? So you need to know what happens in which stage, mostly with the chromosomes, and you need to know the stages in order. Meiosis is different than mitosis, and how is it the same as mitosis, right? So we're kind of building on what we know about mitosis, and you're learning about meiosis in the context of mitosis, mostly. So let's jump into it. So as you should already know, sexual reproduction, that is reproduction that produces offspring that has a unique combination of the genes from the parents. Another way of saying that is something we've already said, which is if we're talking about sexual reproduction, they are genetically unique, right? Genetically unique. And of course, like I've just said, sexual reproduction depends on meiosis and fertilization. And I'm sure you almost all knew that part, right? Sexual reproduction depends on fertilization. I'm sure we all knew that. But again, like I've been saying, it also depends on meiosis. Another way of saying this third bullet point is it depends on making sperm and egg, which is what meiosis is, and it also then depends on those sperm and egg coming together, which is what fertilization is. So yes, before we jump into meiosis, again, this is the process, this is how we make sperm and how we make eggs. So any questions so far? All right, I like this picture from your book, because again, it says it produces offspring that has a unique combination of the genes from the parents. So if you look at those kids versus the parents, it's a great, some couples don't look like this, but I think it's a great mix where the boy looks like the mom and the dad. You can see both features, right? They both look like that, right? You can see the combination. I'm going to talk about how you can get that combination as we talk about meiosis. But first, before we can actually jump into meiosis, you need a little background information. So we're going to talk about homologous chromosomes. And we've already started this conversation, but now we're going to kind of dig a little bit deeper, get a little bit more specific, and talk about it in the context of meiosis and sexual reproduction. All right. As we've already talked about, I think, the different individuals of a species have the same number of chromosomes. Right? So all humans, by definition, unless there's an issue, have 23 pairs of chromosomes, right? Which means we have 46 chromosomes, right? 23 pairs is 46. Like if I had 23 pairs of shoes, I would have 46 shoes, right? So a human somatic cell, that's what I'm getting at here. Those are the ones that have the 46 chromosomes. And I don't know, depending on how you want to learn it, you might just think, okay, most human cells have 46 chromosomes, and there are some exceptions, and I'll teach you the exceptions. But when I say most human cells, we don't just say most human cells. We give those cells a name. Those are called somatic cells. So basically, every cell in your body, other than sperm or egg, those are called somatic cells. And again, they have 46 chromosomes. And you need to know that number. Because I'm not usually big on numbers, and I'm very specific, and I make sure you need to know certain numbers. And you do need to know that humans have 46 chromosomes, or 23 pairs. So if you look at this picture, you can see, right? 1, 2, 3, 4, 5, all the way down. You get down to number 22, and then right here, the sex chromosome. So that's number 23, right? So you have 23 pairs of chromosomes. And I haven't taught you what a karyotype is, but I'm about to on the next slide. All right. I'm not going to ask you how to produce a karyotype, but this is how your book presents it, so that's how I'm going to present it. By talking about how you do how you produce a karyotype, that might help you remember what a karyotype is. So, to produce a karyotype, which again is just basically pictures, like a microscope picture of your chromosome. You can see your chromosomes all lined up. First of all, they have to break open a human cell in metaphase of mitosis. So let me ask you that. Why would it need to be in metaphase? If we're going to produce these pictures right here of these chromosomes, right? Why? Actually, before we even ask specifically about metaphase of mitosis, let me just ask you this. Why, if we're going to take pictures of chromosomes, do they need to be in mitosis in general? Forget metaphase for now. Just mitosis. Why should they be in mitosis if we want to take pictures of them? Why not take pictures of them when they're in interphase? Any guesses? Okay, what's one of the big differences between a cell that's in mitosis? And again, we're obviously talking about the chromosomes, so let's reword it that way. What's the big difference between the chromosomes when they're in mitosis versus when they're not in mitosis? This is something you'll definitely need to know for the exam. Which, hint, by the way, for those of you who are going to retake the second exam, like I told you last week, there's going to be questions on this second exam where some of the choices are mitosis or meiosis, and that's not the correct answer because that's going to be on the third exam, and some people still chose it. But anyway, if no one wants to guess, I'll say it. All right. So remember, in prophase, the chromosomes have to coil up because remember they need to move to different sides of the cell, and before they can do that, they need to not be in that big squiggly mess. So remember, when they're in interphase, they're in a big squiggly mess, and you cannot choose. You can't pick out one chromosome from the other. But when they're in mitosis, they coiled up like they did in prophase. Therefore, you can see them, right? And again, the karyotype thing, that's not important. I know I'm talking a lot about this right now, but that part's not important. What is important, though, what you should know for this third exam is chromosomes coil up in mitosis, right? So that's just a good reminder. Now, why metaphase? I don't know, just probably because they're lined up, so it makes it easier, right? So you have all your sister chromosomes, or all your. So when they're lined up, they're just easier to pick out. Basically, what happens is you take a picture of the cell and you Photoshop, right? You cut and paste all these little things because they don't line up like this in the cell. They line up straight in the middle in metaphase. So again, it's like a copy and paste kind of situation. Anyway, so they break open the cell in metaphase and mitosis. And again, I'm not going to ask you any of this, but that is a good bullet point to know because outside of the context of karyotypes, you need to know that chromosomes coil up for mitosis. Then they stain those chromosomes with dyes. Otherwise, you wouldn't be able to see them. You certainly don't need to know that for this 100 non-biology major course. But yeah, just so you know, they do stain them with dyes. So all those pictures you saw in that lab last week, those were actual pictures. Those are real pictures. It may have been, on the computer as opposed to actually looking at a slide on the microscope. But those pictures were from slides on the microscope, and they did dye the cells. And then, of course, like I said, they take the pictures with the microscope, and then they arrange them by size, right? So, and this is how it works. Not that you need to know this, but they are numbered, right? From number 1 all the way down to chromosomes number 22, and then also your sex chromosomes. They're numbered, and they go from largest. Questions about this side? All right. Here's a drawing on the left, and again, another karyotype on the right that's kind of been given some color to make it easier to see. So what we're looking at here is, again, there's, like, for example, chromosome number 1, which happens to be the biggest, right? That's a pair of homologous chromosomes. There's one that you got from your mom and one that you got from your dad. Of course, I don't know which is which. That could be the other way around. That one could be mom's or dad's, and that one could be mom's. Doesn't matter. The point is you got one set from your mom, one set from your dad. So then we zoom into this chromosome number 1 and see what it looks like in a drawing. So here's what you learned in mitosis, right? In S phase of interphase, this one chromosome duplicated, right? So now we have sister chromatids, right? Because they haven't separated yet, so they're still stuck together at the centromere. So that one chromosome duplicated. This one chromosome also duplicated. Makes sense? You guys are with me? You remember this from mitosis? So again, in the S phase of interphase, the chromosomes duplicate. So that one that you got from your mom, it duplicated. The one you got from your dad, it duplicated. So now both of your chromosomes, or all of your chromosomes really, have duplicated. Any questions about this slide? All right, the next word for attendance will be this one that I'm circling right now. I'm not going to say it. There it is. That's the next word. Here's a picture of me pointing at it. Take a screenshot. Make sure your screenshot shows your whole screen, right? So I can see, like, if you're on a computer, I can see the time of, the date and the time on the bottom and which tabs you have open. That way I can tell it's your screenshot and not somebody else's. Anyway, let's move forward. Homologous chromosomes. Again, that's what we're talking about. They resemble each other in length and centromere position. Let me back up. All that's saying is, all right, so if we're talking about chromosome number 12, randomly, let's say we zoom in, we open me up, remember, let's make it a little bit simpler. We take a blood sample, we look at my genome or my chromosomes, my karyotype. We do the same to her. And then we look at chromosome number 12 randomly. I'm just picking that number. If we were to look at it, mine will look probably just like hers, about the same length. You can see the centromere for number 12 is right here. Not that you need to know this, I'm just pointing it out. You can see the centromere is right there. It's kind of closer to the top than the bottom, right? Some of them would be closer to the bottom. Some of them are closer to the middle, like right there. Anyway, the point is, if we were to look at any human's chromosomes, unless there's something wrong, generally speaking, if they're the same number, they will have the same position, right? The same length. And more importantly for this semester is the second bullet point right here. They carry genes controlling the same characteristics. This is going to be very important, not just for this chapter, but for the chapters moving forward. Homologous chromosomes carry genes controlling the same characteristics. So if I have a gene for stinky feet on the tip of chromosome number 17, I'm making that up. I don't think there is a gene for that. You could look it up for independent work. I bet actually there are genes that contribute a little bit to that is slightly interesting. Not just stinky feet, but stinkiness in general. But anyway, chromosome number 17. At the tip of that chromosome, I have a gene for stinky feet. Let's assume that's true. If that's true, that means for every other human, unless there's something wrong, including each one of you, if you were to look at the tip of chromosome number 17, you would all have the gene for stinky feet. Now, don't get me wrong, you might not have stinky feet. I actually don't have stinky feet. It's one of the few things I could say. Knock on wood, that might change. But the point is, there are different versions, and we'll get to that later. So again, homologous chromosomes carry genes controlling the same characteristics. Or if I'm not giving you a hypothetical example, the gene for your eye color. Well, the gene for the eye color is in the same place, the same chromosome as is for me and every other human, unless there's something wrong. And you can look that up if you want for independent work. And you'll see what I mean when I say we oversimplify it in this course. But yes, there's a simplified version that says there's a single gene responsible for eye color. And we know where it is, and we know how it works. Anyway, again, repeating myself because your book repeats itself. We humans have 46 chromosomes. Right? And then now I'm going to give you some new words here. We have 22 pairs of matching chromosomes that are called autosomes. And then we have a pair called sex chromosomes. And I already told you that earlier, except now I'm putting it in writing. And I didn't use those words autosomes, right? So again, I'm going to back up, and then I'll come back. So if you're taking notes, don't be upset. I'm going to come back to the slide. Like I said, we have 22 pairs. 22 pairs of autosomes, and then that one pair of sex chromosomes. So in other words, you have sex chromosomes, and then you have everything else. And the everything else we happen to call autosomes. So that's something you need to know too, right? Know the difference between autosomes and sex chromosomes. And also know this. This is going to come up in a couple of chapters. So it definitely will be on the exam. Males have, for sex chromosomes, an X and a Y. And females have 2 X chromosomes. That is how we biologically define sex for humans. There are exceptions. And there's a difference between sex and gender. So you can look that up for independent work if you want. That's kind of pushing it, though, because gender is more of a social science construct than a biological construct. But typical male, XY, typical female, XX. And I say typical, I'm going to give you some exceptions to the rule. But anyway, are there any questions about this slide? Homologous chromosomes carry the same genes, same position on the same chromosome, right? All of our chromosomes are called autosomes except the one pair of sex chromosomes, of which males have an X and a Y and females have an X and an X. And another another independent work topic, if you want to look into it, I think I mentioned this in a previous lecture, is that this is just for mammals, right? Well, obviously humans, which is what we're focused on, but what about other animals? What about reptiles, amphibians, birds? What about not even animals at all? Because yes, plants also have sex chromosomes. Fungi have sex chromosomes. So it's all worth looking into. Any questions? All right. This next bullet point, I assume, is going to be largely a review of things you've been learning your whole life. Except maybe I'm going to throw in some new words, but this is probably concepts you already understand and you've already learned. We're talking about gametes and the life cycle of a sexual organism. So officially, in this lecture, I don't think, definitely not today, but I don't remember all of chapter 8, but I don't think officially in a lecture I've defined the word gametes. Of course, you've been introduced to them in labs. Does anybody remember what a gamete is by any chance? Okay, we'll get back to it then. So the life cycle of a multicellular organism is the sequence of stages from the adults of one generation to the adults of the next. I'm not going to test you about that, but that is a little bit different than what you've probably been taught. So, usually if you were to even ask me, a biology teacher, what's the life cycle of a human? Oh, yeah, it's when you're born to when you die, right? But in the context of this conversation, when we're talking about sexual reproduction, then the life cycle is when we're talking about the adults of one generation to the adults of the next, right? So, for example, when I was an adult and then had my daughter with my wife, right? That would be the beginning of the life cycle. And then when, knock on wood, she has not yet, but when she goes to reproduce, that would be the end of that life cycle in the context of this conversation. Not that I'm going to test you on that, but that's how we talk about it when we talk about mitosis and meiosis. So having two sets of chromosomes obviously is a key factor in the life cycle because, well, as I'm about to teach you, they split up, right? Because hopefully you already know this, and this should make sense even if you've never been taught it, but your sperm or your egg, your gametes, only have half the chromosomes, right? If you had the full set of chromosomes in your egg and they got fertilized by a sperm that also had a full set of chromosomes, then you would have twice the number of chromosomes. And as you're going to learn later in this chapter, having too many chromosomes for humans can be very bad. Speaking of which, that brings us to some words you need to know. Diploid versus haploid. Humans are diploid, meaning all of our body cells, or somatic cells, if you will, that's that term I talked to you earlier. All of our somatic cells contain pairs of homologous chromosomes. Pairs. So the key word there is we have pairs of chromosomes. And the way I always remember that is this prefix here, di, very often means two. All of our somatic cells. Of course, different for our gametes. Our gametes, and that's not written on here, so pay attention to what I'm saying. Sorry, it's a mistake on the PowerPoint. Our gametes, so not all of our other body cells, but our sperm and our egg, they are what's called haploid, and they only have one of each pair of homologous chromosomes. So again, if you look at any cell in your body, you would see 23 pairs of chromosomes, right? You would have one of each, one from your mom, one from your dad. Chromosome number 20, one from your mom, one from your dad, right? That's diploid. That's every cell in your body except your sperm or your egg. If you were to look at them, they would only have one set of each. So yes, you're still going to have the full 23 chromosomes, except there's not going to be a pair of each, right? So if you look at chromosome number 10, and again, we're talking about your sperm or your egg. You got one from your mom or your dad, that's it. Which is what we're going to talk about later, right? That makes it haploid. So are there any questions about this portion of the slide before I talk about the next portion? It's very important. Not necessarily terminology. I'm probably not going to test you and ask you what diploid means or what haploid means, but I'm definitely going to use those words in this chapter, so you need to know what they mean when I use them. So you're 23 pairs of chromosomes. And if you were to look at any one of them, you have one from your mom, one from your dad. If you were to look at your cells, excuse me, your sperm or your egg cells, they're haploid. So of those 23 chromosomes, you're only going to have one, right? One from your mom or your dad. So chromosome number 12, I'm just throwing these numbers out here. You might look at yours, look at one of your sperm or eggs, and it might have chromosome number 12. You might have the version from your mom. Then you might look at chromosome number 13, and you'll have it from your dad. There's no rhyme or reason. We'll talk about that later. But again, if you're looking at sperm and egg, you only have one of each chromosome. Anyway, back to the part where you probably do know. In the life cycle of a sexually reproducing organism, the haploid sperm fuses with the haploid egg, and that's a process called fertilization. That's not a word for attendance. So just because I'm circling it, mean anything in that context. But anyway, again, sperm comes together with egg. That's fertilization. I'm sure you already knew that. Only new term, possibly new for you, is the term haploid, right? Haploid sperm, haploid egg comes together in fertilization. The result, this might be a new word for you, right? When you put the sperm and egg together, you get a zygote. So obviously, if you put a haploid thing that has half the chromosomes, right? If you have something with half the chromosomes and you combine it with something else that has half the chromosomes, then together they're going to have the full set of chromosomes. So zygote is diploid. There is an exam, or excuse me, a question. You know, which of these is correct? So you need to know a haploid sperm fuses with a haploid egg, and that produces a diploid zygote. That question will be missed. I don't mean for it to be tricky. I'm telling you right now it's going to be on the exam. So some of the wrong answers would be a diploid egg and a diploid sperm come together, right? That's not right because it's supposed to be haploid. Or I might say a haploid, an egg and a sperm come together to form a haploid zygote, which is, again, not correct because zygote is diploid. So I'll go ahead and tell you now that question is on the exam. So yes, again, your book kind of reiterates what diploid means right there, right? It's diploid, meaning it has two sets of chromosomes, one from each pair. So chromosome number 1, I'm just making this up, I don't know. When I made my son, perhaps I gave my son, for chromosome number 1, maybe I gave him the dad version that I got, right, as opposed to my mom's version. And maybe my wife gave him his dad's version or her dad's version of chromosome number 1. You don't know. It could have been either one, right? When we got together to make my son, and we're talking about chromosome number 1, I could have given my son my mom's version or my dad's version. My wife could have given him her mom's version or her dad's version. How it works. Any questions about this slide? All right. All sexual life cycles involve an alternation of diploid and haploid stages. That's just kind of, I'm not going to ask you about that. It's kind of like an overview statement of what we just said, right? So first thing I told you was what the human life cycle was in the context of sexual reproduction. Then I told you what haploid versus diploid is. Now I'm putting those two things together to just say, all right, if we're talking about the entire life cycle, which again is the adult of one generation to the adult of the next, we're alternating between diploid and haploid. Like right now, we are diploid individuals that have haploid cells in us. And then, of course, this is not a test question either. This is more like a statement that explains why we're learning this. But producing haploid gametes by meiosis keeps the chromosome number from doubling in every generation. Right? Again, like I said, when you have a sperm and an egg come together, if they don't have half the chromosome numbers, then they're going to double, right? So if I didn't go through meiosis and my wife didn't go through meiosis, then our son would have 46 pairs of chromosomes, right? Not 23 pairs, but 46 pairs of chromosomes. And then when he had a kid, then that would double. And then when they had a kid, that would double, right? We don't want to do that. We want each generation. We want to start back at square one. So we need to cut our cells, our chromosome numbers in half for our sperm and our egg. That's what this is all saying. Here's a picture of what we've all said, right? We have some adults. They're going to get together. He's going through meiosis. She's going through meiosis. He's producing egg. She's producing sperm, right? So at this point, they are diploid, but they are producing these haploid versions of themselves, so to speak. They're producing these haploid cells, these haploid gametes. They come together in a process that's called fertilization. At this point, we are left with a diploid zygote, right? That's when you were one cell. And then that thing just keeps going through mitosis over and over and over again until you have a full-blown human being. And I'll go ahead and tell you this. This is maybe on the study guide, but I'm not going to talk about these numbers. For a 100 level non-biology major course, I like to not deal with n's because it could get confusing. Basically, what that means is, right, n equals 23, as you can see up there at the top. We have 23 chromosomes, right? Sort of. 23, but 46 total, 23 pairs. That's all that means, right? And this is how it would get confusing if we try to talk about it. So again, when you see that for this exam, or even reading anything in this semester, you can ignore the 2n equals 46 or n equals 23. One of you did a really good paper about, I think it was raccoon chromosome numbers. Anyway, I read your sources you sent me, and they were talking about the n equals and 2n equals and all that stuff, which I would assume may not have made sense to the person who wrote that independent work, but there you go. That's what it means if you saw those things and didn't know. Let's see. Yeah. Any questions about this slide? All right. So this is kind of an overview of meiosis before we really jump into it in detail. So here we have a pair of homologous chromosomes, right? So again, we have 23 pairs of them. So pick a number in your head, whichever, it doesn't matter. But we can assume that's chromosome number 1. Maybe it's chromosome number 22. Whatever. We don't know. Maybe it's a sex chromosome. Point is, we have a pair of them, right? This is just one chromosome. Don't know which one. We have one that we got from our mom, one that we got from our dad. That's interphase. Then we hit S phase of interphase, where our chromosomes duplicate, right? So let's just say the one on the top is the one from your mom, the one on the bottom is the one from your dad. Versus red, but not that color in nature anyway. We're just using this to differentiate. So the chromosome you got from your mom, it goes through S phase, it duplicates itself, so sister chromatids. The chromosome you got from your dad, it goes through S phase of interphase, it duplicates itself. There you go, now you have sister chromatids. So we still have homologous chromosomes, right? This is still chromosome number 1 or whatever number you may want to give it, right? You still have a version from your mom and a version from your dad, but now they've duplicated, right? Not only they're homologous chromosomes right now, but they also sister chromatids. And it is important for the exam, first of all, to understand the question between a homologous chromosome and a sister chromatid. So remember, a homologous chromosome, that's the pair of chromosomes that happen to be the same number because they have the same genes, right? You got one from your mom, one from your dad. That's what a homologous chromosome is. And remember, sister chromatids are the result of a single chromosome going through S phase and duplicating itself. And of course, if you remember in mitosis, the sister chromatid is split, but we haven't gotten there yet, right? So we have this one single chromosome from your dad, and of course one from your mom. They went through S phase, and each one of them individually duplicated themselves. But they're still there together, except now they are there as duplicated versions of themselves. Any questions so far? I would point this out. Hopefully this doesn't muddy the waters for you, but technically this picture is inaccurate, right? Because technically if we're in interphase, the chromosomes aren't all nice and coiled up where you can see them like that, right? Technically right now they're in a big squiggly mess. But to be able to show what happens, the people who make these drawings make them condensed. Anyway, let's move forward. So here we go. Meiosis. And again, We're just not scratching the surface. We're going to get into detail. I go ahead and tell you right now, one of the differences between meiosis and mitosis, and you do need to know the differences, a lot of the exam will be about that. One of the differences between meiosis and mitosis is in meiosis we have 2 rounds. You ended with 2, end of story. Here, we do it twice. In the first round, we split them up, just like we do in mitosis, except instead of saying split it up or split them up, now I'm going to use specific terms, and you're going to need to understand this. So let me ask you this, because the answer is right in front of your face. Please don't feel shy. Shoot out a guess. What's separated here? Did the sister chromatids separate, or the homologous chromosomes separate? And again, don't be afraid to get the wrong answer, because if you get the wrong answer, I feel like the picture is just not making it clear enough. I don't blame you. You're learning this. Someone else drew this to teach you. So if you don't get it, they just didn't draw it properly. So what's separated? Homologous chromosomes or sister chromatids? For round 1 of meiosis, right? Which one's separated right here? No guesses? The homologous chromosomes separated. See? Here we have sister chromatids. That one just kind of went this way. Those sister chromatids went this way. Those went that way, right? So these sister chromatids did not separate. The sister chromatids you have from your dad's chromosome are still together. The sister chromatids you have from your mom's chromosome, they're still together, right? The sister chromatids haven't separated. Only the homologous chromosomes are separated. And that is definitely one of the big distinctions that you need to know as far as the difference between meiosis and mitosis. In meiosis, we separate homologous chromosomes, at least for meiosis 1, right? But in mitosis, we don't notice. I didn't even teach you about this word homologous chromosome when we talked about mitosis. That was a discussion that we didn't have until we started talking about meiosis. So in mitosis, we only talk about sister chromatids. If there's any questions on the exam that have anything to do with homologous chromosomes, the answer is meiosis. Because only in meiosis do we talk about separating homologous chromosomes. Because that's the only time they ever line up together. The homologous chromosomes, they line up together in meiosis, not mitosis. So anyway, there we go. Round 1, we have separated the homologous chromosomes. So now we have 2 cells. You have 1 cell, you have 2. You've got 1 cell that has a pair of sister chromatids from your mom, right, for chromosome number 1, if you want to call it that. And then you have another cell that has the other version, right, the one you got from your dad, and it's sister chromatids. So again, in meiosis 1, we separate homologous chromosomes. What do you think we're going to separate in meiosis 2? And there's only one choice left. Yes, great. Sister chromatids, thank you. You definitely need to know that. So in meiosis 2, we finally separate the sister chromatids. So what you're left with at the end of the day, when you start with 1 cell, 1 diploid cell, you now have 4 haploid cells, right? So in the first round, we separate the homologous chromosomes. In the second round, we separate the sister chromatids. And essentially, especially as far as the exam is concerned, essentially meiosis 2 is the same as mitosis. And we will get into that a little bit more, but as far as exam questions are concerned, you might as well consider mitosis and meiosis 2 the same. So we're just separating sister chromatids. When there are some differences, but I'm not going to ask about those differences. There's a lot of test questions on here. And we haven't even really gotten into details. So of course, the details don't really matter now that I'm thinking about it. This is the big stuff. This is meiosis 1. What this doesn't show you is from going from here to here, we have prophase, metaphase, anaphase, telophase. Boop. And then from meiosis 2, we're getting here to here, we have prophase, metaphase, anaphase, telophase. It just doesn't show all those things. But they're essentially the same, except in meiosis 1, or excuse me, let me back it up even more. Remember, this whole thing that we talk about is the story of the chromosomes generally. So in mitosis, we just talk about those sister chromatids separating, and that's the end of the story. In meiosis 1, we talk about separating the homologous chromosomes. And then, once they're separated, then we can move on and separate the sister chromatids. So let's see. The next word for attendance will be pair, like a pair of shoes, or a pair of dice, or a pair of chromosomes. Again, if you're watching the video, take a screenshot, make sure it shows everything. I need pointing at the word pair. There you go. All right, if there's no questions about this side, we'll move forward. Yep, and let's actually talk about the process of meiosis. Even though we just did it, let's do it again with the proper words in front of our faces. Meiosis, as I'm sure you already know, and I'm probably not going to actually ask you the definition of meiosis. You just need to know the differences between meiosis and mitosis. But again, meiosis is a cellular division that produces haploid gametes, right? Another word of saying it makes sperm and it makes eggs with half of the chromosomes in diploid organisms. And it resembles mitosis, but has two major differences. So these are all the things I've already told you, except now you're going to see it in writing. The first major difference, and you need to know this for the exam, is that the chromosome numbers are cut in half. So if we take any one of your somatic cells that has 46 chromosomes, right, 23 pairs of chromosomes, if it goes through mitosis, then those resulting two cells are also going to have the original number of chromosomes. But in meiosis, they're cut in half. Well, we already said that. We just didn't say it in this context. We said it in this context, like the first main bullet point, which is that we have haploid gametes, meaning we have half the chromosomes. So yes, in meiosis, the chromosome number is cut in half. The book points it out yet again. Obviously, it's important. How does this happen? Like I've already said, the cell undergoes two divisions, right? So think about it. In mitosis, in S phase, everything doubles, right? All your chromosomes duplicate, and you have sister chromosomes. So double something and cut it in half. What are you left with? The original number. That's mitosis. In meiosis, you double it. Half the original number. Makes sense, right? So again, mitosis doubles the number, cuts it in half, so you're back to the original number. Meiosis, which is what we're talking about now, doubles the number, cuts it in half once, so we're back to the original number, then cuts it in half again, so we're now down to half the original numbers. That is the first major difference between meiosis and mitosis. And again, everything on the slide right now, I've already told you, but now it's there in writing. And worded slightly different in some cases. Any questions about this slide? All right, second major difference is a whole new concept. It's the first time I'm telling you about it. Something called crossing over. Crossing over does not happen in mitosis. It's very important. Without crossing over, we wouldn't be where we are. And it also has important implications in the semester after this chapter. So this will not be the last time we talk about crossing over. What is crossing over? It is the exchange of genetic material between homologous chromosomes. So let's just call this, I don't know, chromosome number 5. You got one from your mom. You got one from your dad. When they're close together in this process, they literally swap chunks. So you can see here, that one's all mom, that one's all dad. And then they get a little bit close together, and what you're left with now is you have one that's all mom with a chunk of dad, and then you have one that's all dad with a chunk of mom. You guys can see that, right? So remember, all those eventually are going to get separated. So you're going to have a cell, the sperm or egg cell, that only has this one. You have another sperm or egg cell that only has that one. So even though all of your chromosomes are going to be separate, you're going to separate the ones you got from your mom versus the ones you got from dad. You're going to separate them. But and even your dad chromosomes are going to have mom information. Make it more complicated. Your book doesn't even talk about this. First of all, sometimes crossing over doesn't happen. And also, it doesn't always happen the way it's shown. It could be a smaller chunk than that. It could be a bigger chunk. It might happen at the bottom and the top, right? So there's so many different ways crossing over can happen. So make sure you understand what crossing over is. Call it playing chromosome footsie. Your mom and dads, they got together, they're playing footsies. But in this situation, they ended up swapping feet. So any questions about crossing over for now? Again, we will talk more about crossing over. Oh yeah, another question that will probably be on the exam when we're talking about crossing over. When does it happen? It happens during the first prophase of meiosis. Now the part that should be easy for you to remember for that is this part right here. The first, right? Which should make sense, because if you know meiosis, which I don't expect you to yet, but you will after you study it and let this sink in. But remember, you start off with homologous chromosomes, and then meiosis 1, you separate those homologous chromosomes. So when you start meiosis 2, those homologous chromosomes are in completely different cells. So there's no way crossing over could happen in the second prophase or the second anything, because the homologous chromosomes are separated in that first round of meiosis. So to me, the most important thing is the easiest thing to remember is that it happens during the first meiosis, not the second one. The harder part, and this I shouldn't say always, but this sometimes tripped me up as a young student, is that it happens in prophase. To me, I don't know why, but my brain just always wanted them to be in metaphase. Actually, I do know why, because the metaphase are all lined up together. So to me, that makes sense. Like, hey, they're lined up. But yes, the homologous chromosomes, this whole meiosis 1, they are together. They're not lined up in the middle yet necessarily, but they're together. So sometime during the first prophase of meiosis, that's when crossing over occurs. And honestly, even though there might be a test question about it, I feel like that's less important information. You can understand everything else I'm teaching you this semester without understanding exactly when crossing over happens. The only reason I'm even talking so much about the fact that it happens during the first meiosis is because what is important is that you understand that the homologous chromosomes get separated during the first round of meiosis. So by that logic, which was something you need to know separately, by knowing that, you should know that this could have happened in the second meiosis. Not because I'm going to ask, not because it's important for understanding crossing over, but again, you just need to understand in meiosis 1, we separate homologous chromosomes. In meiosis 2, we separate sister chromatids. Any questions about this slide? Here's a picture, some drawings of meiosis. Here we are in interphase. This should look exactly the same as it does in mitosis. Right? We have the centrosomes that is hanging around. They haven't done anything yet. Specifically, they haven't formed that mitotic spindle that's going to be moving around the chromosomes. We have this big squiggly mess of chromosomes. You can't pick out one chromosome from the other in there, right? It's a big squiggly spaghetti incident. You move forward, that's interphase. And then you jump into prophase 1. And look, this is exactly the same as it is in mitosis with some exceptions. So this is why I'm not going to ask, okay, for meiosis, what happens in prophase 1, prophase 2, prophase 1, metaphase 1, anaphase 1, and telophase 1. I'm not going to get that specific because other than the fact that we're separating homologous chromosomes as opposed to sister chromatids, other than that, things are pretty much the same. What happens in prophase? Well, the mitotic spindles form and connect to the chromosomes because the chromosomes need to be moved around. The nuclear envelope needs to disappear because we're moving the chromosomes on different sides of the cell. The chromosomes need to condense up, again, because we're going to move them across to the other sides of the cell. So this is the same thing as it is in mitosis. What happens in metaphase? Close to the same things that happens in mitosis, right? All the chromosomes lined up in the middle. Except in this case, the homologous chromosomes are being lined up in the middle, not the sister chromatids. And what's happening right here in metaphase, like I said, they've lined up. There's a tug of war going on. What happens when that tug of war breaks them and separates them apart? At that point, that is technically called anaphase because they have separated, just like in mitosis, except now what has separated? The homologous chromosomes are separated, not the sister chromatids, but the homologous chromosomes. What happens in telophase? Basically, again, the opposite of prophase. We'll just leave it at that, right? Except, again, big picture here. In metaphase, meiosis 1, we didn't separate sister chromatids. We separated homologous chromosomes. Everything else is the same. So for the exam, for mitosis, I'm going to say in this stage, the chromosomes did what? In this stage, the chromosomes did what? In this stage, you already answered those questions for the mitosis, and they're essentially the same. The question for meiosis is going to be, in which is meiosis 1 or meiosis 2, in which one of those are the sister chromatids separated or are the homologous chromosomes separated? And the answer is, if we're talking about homologous chromosomes, That's meiosis 1. So any questions about that? All right, meiosis 2. Here, now we started with 2 cells because, we started with 1 in meiosis 1. We split that into 2. So now we're coming into this with 2 cells. Same thing. I'm not going to go through it. Prophase, again, they're condensed, all the things I said. Metaphase, they met in the middle. Anaphase, they separate. Telophase, they're completely separated, and we have cytokinesis. So again, same thing. Again, for the exam, meiosis 1 separates homologous. Now is essentially identical to mitosis, and it separates sister chromatids. And again, a big thing, and I think this should be on the next slide. So the next slide, I think, is all this in one picture. If it's not, I'll back up. Yeah, I'll back up then. So remember, also, in meiosis, we started with 1 chromosome. We ended with 1, 2, 3, 4 genetically unique daughter cells. Unlike in mitosis, which is not seen here, but in mitosis, we start with 1 cell. We end with 2 genetically unique chromosomes. Cell, we end with 4 haploid cells. In mitosis, we start with 1 diploid cell and end with 2 diploid cells. All right. Any questions about this slide? Okay. There's a picture of meiosis 2 in the Lilly cell. It's perfect. This really is perfect. So when we get back from spring break, after you've probably purged all this, and I don't blame you because that's what spring break is partially for, we're going to come back and review everything we said before we jump into chapter 9. So does anybody have any questions about anything? All right. I don't expect anybody to work over spring break. Enjoy yourself, but I just want you to know I will be. So if you need me, you feel like you want to use that time to catch up or whatever it is, I'll be here for you. Just let me know. And I do expect to have all the labs graded for photosynthesis and respiration. So coming back from spring break, you'll be able to start focusing on lab reports. There we go.